Welcome students. Today we will talk about adaptation of roots for reproduction and interaction with microbes. Roots are that part of a plant which normally develops beneath the surface of soil and are principal water absorbing organ of a plant. They vary widely in their morphology, exhibit structural and developmental differences correlated with more or less pronounced physiological specializations. Roots with obvious function or structure are described as storage, fleshy, fibrous, aquatic, tap, aerial, and holdfast. Environmental conditions often influence the root system. Plants growing in dry soils usually have better root systems. Many plants growing in sandy soils develop shallow, horizontal, lateral roots which spread out close below the surface of soil over a distance of tens of meters. For example, in tamarix and tama. Two types of roots are being recognized on the base of their origin. First, tap roots and second, adventitious roots. Tap roots develop from the apex of the embryo that is destined from its origin to give rise to a root and from the pericycle of relatively mature parts of roots. Most dicotyledons and gymnosperms possess a root system based on the taproot and its branch. The taproot produces the lateral or branch roots in an acropetal sequence. The taproot is often called the primary root. Branches of the first order, the secondary roots, and branches of the second order, the tertiary roots. The root systems of monocotyledons are composed of adventitious roots. The term adventitious roots is used for roots that originate from the stem, branch, leaves, old woody roots, rhizomes, or from callus of cuttings. Special importance has been given to those adventitious roots that develop from the callus or cuttings. Adventitious roots are important in plant propagation, particularly in rooting of stem cuttings and the generation of root systems on root cuttings or root grafts. Propagation by roots is a form of vegetative propagation or asexual reproduction of a plant. It does not involve flowers, pollination and seed production. Only one plant is involved and the offspring is the result of one parent. The new plant is genetically identical to the parent. Now, adaptation of roots for plant propagation. Roots undergo many modifications to adapt themselves for plant propagation, like development of buds on roots. The roots of some plants develop adventitious buds on them, for example, shisham, guava, muraya, albizia lebec, etc. The formation of buds on roots make possible the propagation of plant by root cuttings and is also an important means of spread of noxious weeds. Buds develop on roots of various ages and structure. They frequently arise endogenously, like lateral or adventitious roots. Endogenous origin was observed in naturally occurring or naturally growing roots and in isolated roots also. The bud may originate in the pericycle of a younger root and be at first deceptively similar to a root primordium. In an older root, it may be found in a callus-like proliferation of ray tissue, giving rise to more than one bud, or it may be initiated exogenously in the callus-like growth, derived from the phylogen. Buds often arise near lateral roots, and if these or their traces are still alive, the buds may become connected to the lateral root trace. If a bud arises in the pericycle, the vascular connection with the initiating root is formed by acropetal differentiation. But if the bud is initiated near the surface, the vascular differentiation is basipetal. Now, tuberous roots or fasciculated roots. Tuberous roots or fasciculated roots are an underground storage organ formed from a taproot. may become swollen with food storing tissues as in Dacus, Pasternaca, Brassica napus, and Raphanus sativus. Together with buds at the base of the old stem, just above the taproot, they form organs of perennation and vegetative propagation. The enlarged area of the root tuber or storage root can be produced at the end or middle of the root or involve the entire root, as in the Ipomia batatas, cassava, yams, dahelia, asparagus, and Hemerocallis fulva. Root tuber is different in origin but similar in function to a stem tuber. In root tubers, there are no nodes and internodes or reduced leaves. Root tubers have proximal and distal ends. The former is attached to the old plant and has crown tissue that produce buds which grow into new stems, and the later normally produce unmodified roots. In stem tubers, the order is reversed, with the distal end producing stems. Tuberous roots are biennial in duration. In the first year, the plant produces root tubers, and at the end of the growing season, the plant shoots often die, leaving the newly generated tubers. In the next growing season, the root tubers produce new shoots. As the shoots of new plant grow, the stored reserves of the root tubers are used in formation of new roots, stems, and reproductive structures. Any remaining root tissue dies concurrently to the plant's regeneration of next generation of root tubers. Plants with root tubers are propagated in late summer to late winter by digging up the tubers and separating them, making sure that each piece has some crown tissue for replanting. Such roots can also be called stalks because they can be stalked for sowing in the next season. Now, root sprout or sucker. The roots of most plants produce cytokinins. As these accumulate in the roots, the cytokinins induce shoot formation. Near the base of the shrub, a new shoot will begin to grow. That travels underground for some distance, turns up at its end and produces a new plant. The new plant is called a root sprout or sucker, and the process is called suckering. The suckering is a form of budding responsible for reproduction of regeneration of a plant by shoots that arise from an existing root system. Species that characteristically produce suckers include ulmus, Taraxacum, red raspberries, and members of the rose family, Rosa. In wetlands of Connecticut, the spice bush, Lindra benzoin, and sweet pepper bush, Clathra alnifolia, propagate naturally this way. Contractile roots. The formation of one corm on top of another tends to bring the successive corms nearer and nearer to the soil surface. Adventitious roots develop from the base of the new corm. Once these have grown firmly into the soil, a region near their junction with the stem contracts and pulls the new corm down, keeping it at a proper level within the soil or on its surface. These special roots are termed as contractile roots. Such roots are known to exist in many herbaceous dicots. For example, Medicago sativa, Daucus, Trifolium, Oxalis, etc., and in many bulbous and cormous monocots. For example, Gladiolus, Ferranasa chloracra, etc., which counteract the tendency in successive generations to grow out of the soil. As each bulb is pulled a little further into the soil, and additional contractile roots are developed each year. When a region of stable temperature is reached, the contractile roots quit pulling. Taraxacum also have contractile roots, and their presence is noticeable because the lower leaves may look like they are coming right out of the ground. In reality, the roots are pulling the stem downwards. Thus, contractile roots play a very important role in keeping the new corms and bulbs at a proper distance below the soil surface. It is quite obvious that by the superposition of new corms on the top of the old ones, year after year, there would be the possibility of coming these structures through the surface of the ground, thus exposing them to temperature stress like cold and frost and rendering them useless. On the contrary, we see that these structures are buried deeper in the ground, deeper than their parents, and are thus kept at a temperature higher than the temperature immediately at the soil surface. Now, propagation by root cuttings. Propagation by root cuttings is an artificial method of vegetative propagation. Root cuttings are taken from the young plant stalk during the winter and spring months to ensure that they are saturated with stored foods. This time frame also prevents cutting during the time the parent plant is rapidly expanding shoot growth. Cutting during active expansion will take food stores away from the root system. Another consideration when dealing with root cuttings is to make sure that the polarity of the root is correct before planting. The portion of the root that was located nearest the crown of the plant should be planted up. Within 3 to 4 weeks, the root cuttings develop roots and occasionally some small shoots. This technique is useful in propagating hard woody plants and forestry. Now, plant microbe interaction. Plant roots exude an enormous range of potentially valuable small molecular weight compounds into the rhizosphere. Some of the most complex chemical, physical and biological interactions experienced by terrestrial plants are those that occur between the roots and their surrounding environment of soil, that is the rhizosphere. Interactions involving plants' roots in the rhizosphere include root-root, root insect and root microbe interactions. These interactions may be classified as positive and negative associations. Interactions between plants and soil organisms are important for understanding a range of intrinsic processes such as nutrient cycling, ecosystem functioning and carbon sequestration. On the basis of current knowledge, plant microbial interactions can be classified into 3 basic groups. First, negative or pathogenic interactions, second, positive interactions, and neutral associations or neutral interactions. Symbiotic and pathogenic interactions have attracted the most scientific attention. Positive interactions include symbiotic associations with mycorrhizal fungi and root colonization by bacterial biocontrol agents and plant growth promoting bacteria, that's PGBB. Negative interactions include competition or parasitism among plants, pathogenesis by bacteria or fungi, and invertebrate herbivory. Plant microbe interactions can positively influence plant growth through a variety of mechanisms, including fixation of atmospheric nitrogen, increased biotic and abiotic stress tolerance imparted by the presence of endophytic microbes, and direct and indirect advantages imparted by plant growth promoting rhizobacteria. Bacteria can also positively interact with plants by producing protective biofilms or antibiotics, operating as biocontrols against potential pathogens, or by degrading plant and microbe produced compounds in the soil that would otherwise be allelopathic or even autotoxic. However, rhizosphere bacteria can also have detrimental effects on plant health and survival through pathogen or parasite infection. Now, positive plant microbe interactions. The symbiotic association between certain plant and microorganisms play an important role in soil fertilization and improves their growth and mineral nutrition. Microorganisms implicated in these symbiotic interactions are from 2 groups. First, bacteria, commonly rhizobium, and second, fungi, commonly called mycorrhiza. In rhizobium type, a special type of swelling appears on the surface of the root called nodules as a result of penetration of nitrogen-fixing bacteria in the root cortex. In mycorrhizal association, fungal group is involved in the uptake of nutrients with low mobility, for example phosphorus, and also other nutrients like copper and zinc. Nodulation of legumes by Rhizobia. Rhizobia is the name given to the group of genera of alpha proteobacteria, commonly belonging to the family Rhizobiaceae, which includes all of the nitrogen fixing species that produce nodules with legumes like Allorhizobium, Azorhizobium, Bradyrhizobium, Mesorhizobium, Phylobacterium, Rhizobium and Cynorhizobium, as well as the plant pathogen Agrobacterium. Rhizobia and their legume hosts must recognize each other for nodulation to begin. Different species of Rhizobia are specific to their host plants, but often can infect more than one plant. For example, Rhizobium leguminosarum forms nitrogen fixing root nodules on plants of pea, that's Pisum sativum, and lentil, that's Lens culinaris. Nodulation is a process of complex interactions between the partners. First of all, the molecular mechanisms of recognition between the host plant and the bacteria symbionts are considered as a form of cell-to-cell interorganismal communication. A precise exchange of molecular signals between the host plant and Rhizobia over space and time is essential for the development of effective root nodules. The first apparent exchange of signals is involved in the secretion of phenolic compounds like flavonoids or isoflavonoids by host plant. These specific plant exudates not only induce plant's nod gene expression but also rhizobial chemotaxis. Now, nitrogen fixing organisms. Organisms that can fix nitrogen, that convert the stable nitrogen gas in the atmosphere into biologically useful form, all belong to a biological group known as prokaryotes. These organisms have the ability to reduce dinitrogen to ammonia, accompanied by reduction of protons to hydrogen, with the aid of enzyme complex nitrogenase, a process known as biological nitrogen fixation. Reducing power is provided by NADPH ferredoxin, that's reduced ferredoxin, through an iron molybdenum center. The enzyme nitrogenase catalyzes the conversion of atmospheric gases dinitrogen to dihydrogen to ammonia, like nitrogen plus 3 times hydrogen gives ammonia. This reaction seems simple enough as the atmosphere has 78% nitrogen, but the incredibly strong, that's triple bonds in nitrogen, makes the reaction very difficult to carry out efficiently. In fact, nitrogenase consumes approximately 16 moles of ATP for every molecule of nitrogen to reduce into ammonia, which makes it one of the most energy expensive processes known in nature. Another striking property of nitrogenase is its sensitivity to oxygen. This enzyme gets denatured on exposure to oxygen. For effective reduction of nitrogen to ammonia, an enzyme called the leg hemoglobin is produced by roots of the legumes in response to infection with Rhizobia. It buffers the concentration of free oxygen in the cytoplasm of infected plant root cells to ensure the proper function of root nodules. It has a high affinity for oxygen. This allows an oxygen concentration that is low enough to allow nitrogenase to function, but high enough so that it can provide the bacteria with oxygen for respiration. Now, the Rhizobium legume association. Rhizobium legume symbiosis begins with two free living organisms and ends with intimate cellular coexistence. Rhizobium legume symbiosis is highly host specific. Rhizobium bacteria recognize a specific plant, provoke development of a root nodule and invade the plant tissues. Eventually, the rhizobium cells transfer themselves into a host cell, surround themselves with plant membrane and arrange nutrient exchange with plants. Now, steps involved in the nodule development process. The bacteria encounter root, they are chemotactically attracted towards specific plant chemicals, that's flavonoids, exuding from root. Second, once attracted to the root, bacteria attach themselves to the root hair surface and secrete specific oligosaccharide signal molecules, nod factors. The next, in response to oligosaccharide signals, the root hair becomes deformed and curls at the tip. Bacteria become enclosed in a small pocket. Next, cortical cell division is induced within the root. Then infection thread penetrating through several layers of cortical cells and induce a proliferation of its cells. Cells in advance of the thread divide and organize themselves into a nodule primordium. The branch infection thread enters the nodule primordium zone and penetrates individual primordium cells. The bacteria released from the infection thread into the cytoplasm of the host cells but remain surrounded by the peribacteroid membrane. Failure to form the peribacteroid membrane results in the activation of host defenses and or the formation of ineffective nodules. Infected root cells swell and cease dividing. Bacteria within the swollen cells change form to become endosymbiotic bacteroids which begin to fix nitrogen. Now next, mycorrhizal association. The term mycorrhiza was coined by A.B. Frank, a scientist in Germany, more than 100 years ago. It literally means fungus root and describes the mutualistic association existing between a group of soil fungi and higher plants. The association is based on the plant component providing carbohydrates and other essential organic compounds to the fungi. In return, the fungal component, which colonizes both the root and the adjacent soil, helps the plant take up nutrients by extending the reach of its root system. Fungi become integrated into the root structure, or fungi lives in close association with plant roots. It means that fungal hypha may live on the external surface of the root as ectomycorrhizal association or may invade root cells as endomycorrhizal associations. Now, endomycorrhizal associations. In the endomycorrhizal associations, the fungi forms an inconspicuous mycelium on the root surface but invades the interior of the root cells, that's cortical cells, and produce characteristic vesicles which are membrane bound organelles of varying shapes inside or outside the cortical cells, and orbuscules which are clusters of finely divided tree-like hyphae. These mycorrhizae are thus called vesicular arbuscular mycorrhizae or VAM type. Orbuscules are believed to be the sites where materials are exchanged between the host plant and the fungi. Vesicles generally serve as storage structures, and when they are old, they can serve as reproductive structures. Vesicles and orbuscules, together with large spores, constitute the diagnostic feature of the VAM mycorrhizae. These occur in Orchidaceae, Ericaceae, Acer, Liriodendron, etc. Certain mycorrhizae have been called ectoendomycorrhizae because of the occurrence of penetration hyphae of ectomycorrhizal type into the host cells. Both VAM fungi and ectomycorrhizal fungi extend hyphae from the root into the soil, and these external, that is extra radical hyphae, are responsible for translocating nutrients from the soil to the root. Mycorrhizal fungi and fungi generally have a strong influence on soil structure. Their hyphal strands help to hold soil aggregates together, and they also excrete organic substances that help cement the aggregates. Hyphae conduct water and immobile nutrients like phosphorus to roots despite disruption of capillary water flow in soil. Now, benefits of ectomycorrhizae and vesicular arbuscular mycorrhizae. Now, it provides drought resistance, pathogen resistance, enhances nutrient uptake, that is phosphate and potassium, enhanced nutrient uptake like combined nitrogen, phosphorus, zinc and copper, increased tolerance to pH change and increased root growth. Now benefits to the fungi. First, access to the plant exudates, and second, direct benefit from trees' photosynthetic activity. Thank you. This concludes today's topic, that was about adaptation of roots for reproduction and their association with microbes. So in this topic we studied how roots modify themselves to be used as a means for propagation, and we also studied about various interactions that roots undergo with microbes for the benefit of the microbes as well as for the benefit of the plant.